Welcome back to one more video, guys. In this video we are going to discuss about malaria, infection, life cycle and pathogenicity. Let us briefly describe this life cycle of malaria parasite. Malaria means usually bad air. The people used to assume that this might spread through air. This parasite was discovered by Charles Laveran, and Sir Ronald Ross discovered oocysts in the crop wall of mosquito, for which he received Nobel Prize. This he done in the Secunderabad, for which every 20th August we celebrate World Mosquito Day because of his discovery. And 25th April we celebrate the World Malaria Day, which is regarding the research condition. The causative agent of malaria is genus Plasmodium, which belongs to the sporozoa. It has basically four major genuses: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, and Plasmodium malariae. Vivax causes benign tertian malaria, falciparum causes malignant malaria, ovale causes mild malaria, and malaria causes the quartan tertian malaria. In this video, we'll discuss about the life cycle of vivax. It is responsible for killing Alexander the Great, which you all know. Here, falciparum is associated with brain disease, and Negroes are usually resistant to falciparum because of sickle cell anemic blood. Let us see the structure of the sporozoite, which is infective stage. Here, the spore means seed and zoa means animal, the spore-like animal. It is an elongated shape pointed at both ends. At the anterior end, it contains a depression called apical cup, which contains two glands which are secretory in function, and these secrete cytolytic or proteolytic enzymes. At midist, it contains nucleus attached with endoplasmic reticulum. Also, it contains mitochondria, which deals with the aerobic respiration. Also contain Golgi complex. In the longitudinal section of the body, it contains the elongated microtubules, which help in the wriggling movement of this sporozoite. Entire sporozoite, when you see, discovered by a proteinaceous pellicle, which gives its protection. It is a sickle shape, measuring about 15 microns in length and 1 micron in diameter. So that's how the sporozoite. When such sporozoite in mosquito saliva, if it bites to us, nearly 2,000 of such sporozoites can enter our bloodstream and can cause malaria. Let us see the life cycle of the Plasmodium vivax in detail. It is a digenic intracellular parasite. Digenic means two hosts. The primary host is female Anopheles mosquito. Secondary host is man. It is intracellular in hepatocytes and RBC. In hepatocytes, it performs hepatic schizogony, which is a division, and RBC it performs the erythrocytic schizogony. Schizogony usually means splitting, and gony means division. Let's see the hepatic schizogony. This cycle is a liver cycle discovered by Short and Graham. Whenever the primary host, the female Anopheles mosquito, which is infected with the sporozoite, it can bite to us. You have to remember about 460 species are recognized to transmit the genus Plasmodium to us. And only females bite, just remember. With the proboscis, the mosquito can find the nearby blood vessels for the inoculation of the sporozoite. When it bites, nearly 2,000 of such sporozoites can enter into our bloodstream. 2,000 of those sporozoites, and through the bloodstream, the sporozoites can reach the liver, where they can perform the liver cycle in the hepatocytes by feeding the contents of the liver cell. This is how it goes. Now, when the sporozoite enters the hepatocyte by a cytolytic enzyme piercing the hepatocyte, it can transform into a feeding stage called tropozoite, by which it can feed on the contents of the liver cell. The trophozoite will soon transform into a large schizont after feeding on the liver contents, and this condition is called as an hypertrophy. Soon this schizont bursts, releasing the contents of the cell, approximately releasing 12,000 cryptozoites. These are called the first generation merozoites. This entire cycle is called as a pre-erythrocytic cycle, and it lasts about 8 days. Now remember one point. The period between the first entry of Plasmodium in the form of sporozoite and the second entry in the form of cryptozoite is called as a pre-patent period. It lasts about the 8 days. Now these cryptozoites have two options. Either they can enter hepatocytes or they can enter RBC. If they enter hepatocytes, they undergo the similar changes like previous one, transforming again to trophozoites and feeding on the liver content. Soon this trophozoite can transform into a large schizont, but there is a difference now. Now when this schizont undergoes schizogony, it releases two different kinds of merozoites. Now you are going to see the two kinds of merozoites here, the small ones and the big ones. The big ones are called macrometacryptozoites and the small ones are called the micrometacryptozoites. These big ones enter into hepatocytes and continue the cycle, the exoerythrocytic cycle. And the small ones enter into RBC here and they perform the erythrocytic cycle. Erythrocytic cycle here is discovered by the Golgi and it's called the Golgi cycle. Now remember one point. This cycle is initiated either by the cryptozoites of pre-erythrocytic cycle or the micrometacryptozoites of the exoerythrocytic cycle. When they enter into RBC, they transform into trophozoites similarly, but these trophozoites will soon develop a vacuole, pushing the cytoplasm to periphery. This stage is referred as the signet ring stage because it is looking like a ring. Now the signet, they lose the vacuole and develop the pseudopodia, and this stage is called amoeboid stage. With the help of pseudopodia, they feed on the RBC and develop into a large schizont. Now in this stage, you are going to see some of the weird dots on RBC. These are referred as Schaffner's dots, which are believed to be antigens. But not all the dots are same. For those of the falciparum are called as a malarious dot, for those of malariae are called Zeeman dots, for those of ovale are called James dots. These all are the antigens released by the Plasmodium. Soon the schizont burst and releasing the merozoites approximately 12 to 24. And they again continue the cycle. In this stage, you also see the malaria pigment called hemozoin, which is the damaged portion of the hemoglobin. The cycle entirely lasts for the 48 hours and re-enters. Now here is the point. The period between the first entry of Plasmodium in the form of sporozoite and the first appearance of the malaria symptoms is called as an incubation period. It approximately lasts about 10 to 14 days and it is different for different genus. That's all about the part 1. In next video we'll discuss about the life cycle in the mosquito phase. Thank you.